Hello and welcome to the second video in our course on cell biology and genetics. In this video, we're again going to talk about chromosomes. Last time we talked about what chromosomes are, we talked about their structure, different types, etc. If you haven't seen the video, the link is in the description. Today, we're going to talk about what histones are and what their function is. We are also going to look at the structural units of the chromosome, and I'll explain what chromatin is and how it can change. After watching this video, you will also understand the terms histones, nucleosome, chromatin, linker DNA, histone octamer, heterochromatin, and Euchromatin. By the way, you can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. Okay, so let's start. As we said last time, chromosomes consist of a single molecule of DNA and proteins that modify its structure. This complex of DNA and proteins is called chromatin. And chromatin fiber is also the basic subunit of the chromosome. Chromatin proteins can be divided into two classes, the histones and the non-histone chromosomal proteins. Histones are very important, and for that reason they are present in chromosomes in almost the same amount as the DNA. They form a specific structure of the chromatin which is called a nucleosome. Nucleosome is a basic structural unit of the chromatin. We often describe the chromatin fiber as a series of beads on a string. The string is in fact the DNA, and the bead is a nucleosome. Because the DNA connects the individual nucleosomes, it is also sometimes referred to as the linker DNA. Every nucleosome consists of DNA that is wrapped around it and a complex of histones. It is in fact a complex of 8 histone proteins: H2A, H2B, H3, and H4, all of them present in 2 molecules. This structure of 8 histone proteins is sometimes referred to as the histone octamer. So as we said, a series of nucleosomes forms the chromatin fiber. And the histone that allows this structure to be formed is the H1 histone, and its function is to put the individual nucleosomes close together. So that's it for histones and their function. Now the structure of the chromatin and its condensation can be adjusted, and cells have several mechanisms to do so. One of these are chromatin-remodelling complexes, complexes of proteins that change the position of DNA wrapped around the nucleosome, making the DNA more or less accessible to other proteins in the cell. Cells can also chemically modify histones to change the structure of the chromatin, again making the DNA more or less accessible. The consequence of these mechanisms is that the structure of the chromatin is not the same in every region, in every part of the chromosome. Parts of chromosomes that contain genes that are being expressed are more extended, while regions of chromosomes that contain genes that are not being expressed are more condensed, which means that they are less extended. Regions of the chromosome that are highly condensed are called heterochromatin, and heterochromatin is usually concentrated around the centromere and the telomeres, about which we talked about in the previous video. Regions of the chromosomes that are less condensed are called euchromatin. And as we already said, if we want to express a gene, we want its DNA extended, which means that DNA in the heterochromatin is usually not expressed. Okay? Okay, so this is it for chromosomes. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos about cell biology or medicine in general, please subscribe. Thank you again, and don't forget that medicine can be easy.